Diabetes is also a disorder of fat metabolism. Nowadays, all blood that is drawn from a patient is sent right off to a laboratory to be analyzed. The doctor rarely looks at it, but 100 years ago, the quality and consistency of the blood were valuable guides to diagnosis. Doctors knew that diabetes involved an inability to metabolize not just sugar but fat, because blood drawn from a diabetic's vein was milky, and when it was allowed to stand, a thick layer of cream invariably floated to the top. In the early years of the 20th century, when diabetes had become epidemic and was not yet controllable with any medication, it was not unusual for a diabetic's blood to contain 15 to 20% fat. Jocelyn even found that blood cholesterol was a more reliable measure of the severity of the disease than blood sugar. He disagreed with those of his contemporaries who were treating diabetes with a low carbohydrate, high fat diet. The importance of the modification of the treatment to include control of the fat of the diet is obvious, he wrote. He issued a warning appropriate not only for his contemporaries but for the future. When fat ceases to be metabolized in a normal manner, no striking evidence of it is afforded, and both patient and doctor continue to journey along in innocent oblivion of its existence, and hence fat is often a greater danger to a diabetic than carbohydrate. The linked failure of both carbohydrate and fat metabolism is a sign of disordered respiration in the mitochondria, and the mitochondria we have seen are disturbed by electromagnetic fields. Under the influence of such fields, respiratory enzyme activity is lower. After a meal, the cells cannot oxidize the breakdown products of the proteins, fats, and sugars that we eat as quickly as they are being supplied by the blood. Supply outspreads demand. Recent research has shown exactly how this happens. Glucose and fatty acids proposed University of Cambridge by chemists Philip J. Randall in 1963 compete with each other for energy production. This mutual competition, he said, operates independently of insulin to regulate glucose levels in the blood. In other words, high fatty acid levels in the blood inhibit glucose metabolism and vice versa. Evidence in support appeared almost immediately. Jean-Pierre Felber and Alfredo Vanotti at the University of Lausanne gave a glucose tolerance test to 5 healthy volunteers and then another one a few days later to the same individuals while they were receiving an intravenous infusion of lipids. Every person responded to the second test as though they were insulin resistant. Although their insulin levels remained the same, they were unable to metabolize the glucose as quickly in the presence of high levels of fatty acids in their blood, competing for the same respiratory enzymes. These experiments were easy to repeat, and overwhelming evidence confirmed the concept of the glucose fatty acid cycle. Some evidence also supported the idea that not only fats, but amino acids as well, competed with glucose for respiration. Randall had not been thinking in terms of mitochondria, much less what would happen if an environmental factor restricted the ability of the respiratory enzymes to work at all. But during the last decade and a half, finally some diabetes researchers have begun focusing specifically on mitochondrial function. Remember that our food contains 3 main types of nutrients: proteins, fats, and carbohydrates. that are broken down into simpler substances before being absorbed into our blood. Proteins become amino acids. Fats become triglycerides and free fatty acids. Carbohydrates become glucose. Some portion of this is used for growth and repair and becomes part of the structure of our body. The rest is burned by ourselves for energy. Within ourselves, inside tiny bodies called mitochondria, amino acids, fatty acids, and glucose are all further transformed into even simpler chemicals that feed into a common cellular laboratory called the Krebs cycle, which breaks them down the rest of the way so that they can combine with the oxygen we breathe to produce carbon dioxide, water, and energy. The last component in this process of combustion, the electron transport chain, receives electrons from the Krebs cycle and delivers them one at a time to molecules of oxygen. If the speed of those electrons is modified by external electromagnetic fields, as suggested by Blank and Goodman, or if the functioning of any of the elements of the electron transport chain is otherwise altered, the final combustion of our food is impaired. Proteins, fats, and carbohydrates begin to compete with each other and back up into the bloodstream. Fats are deposited in arteries, glucose is excreted in urine, the brain, heart, muscles, and organs become oxygen deprived. Life slows down and breaks down. Only recently was it proven that this actually happens in diabetes. For centuries, scientists had assumed that because most diabetics were fat, obesity causes diabetes. But in 1994, David E. Kelly at the University of Pittsburgh School of Medicine, in collaboration with Jean-Aimé Simoneau at Laval University in Quebec, decided to find out exactly why diabetics have such high fatty acid levels in their blood. 72 years after insulin was discovered, Kelly and Simoneau were among the first to measure cellular respiration in detail in this disease. To their surprise, the defect turned out not to be in the cell's ability to absorb lipids, but in their ability to burn them for energy. Large amounts of fatty acids were being absorbed by the muscles and not metabolized. This led to intensive research into all aspects of respiration at the cellular level in diabetes mellitus. Important work continues to be done at the University of Pittsburgh as well as at the Joslin Diabetes Centre, RMIT University in Victoria, Australia, and other research centers. What has been discovered is that cellular metabolism is reduced at all levels. The enzymes that break down fats and feed them into the Krebs cycle are impaired. The enzymes of the Krebs cycle itself, which receives the breakdown products of fats, sugars, and proteins, are impaired. The electron transport chain is impaired. The mitochondria are smaller and reduced in number. Consumption of oxygen by the patient during exercise is reduced. The more severe the insulin resistance, i.e., the more severe the diabetes, the greater the reduction in all these measures of cellular respiratory capacity. In fact, Clinton Bruce and his colleagues in Australia found that the oxidative capacity of the muscles was a better indicator of insulin resistance than their fat content, which drew into question the traditional wisdom that obesity causes diabetes. Perhaps they speculated obesity is not a cause but an effect of the same defect in cellular respiration that causes diabetes. A study involving lean, active young African American women in Pittsburgh, published in 2014, seemed to confirm this. Although the women were somewhat insulin resistant, they were not yet diabetic, and the medical team could find no other physiological abnormalities in the group except two. Their oxygen consumption during exercise was reduced, and mitochondrial respiration in the muscle cells was reduced. In 2009, the Pittsburgh team made an extraordinary finding. If the electrons in the electron transport chain are being disturbed by an environmental factor, then one would expect that diet and exercise might improve all components of metabolism, except the last, energy producing step involving oxygen. That is exactly what the Pittsburgh team found. Placing diabetic patients on calorie restriction and a strict exercise regime was beneficial in many respects. It increased the activity of the Krebs cycle enzymes. It reduced the fat content of muscle cells. It increased the number of mitochondria in the cells. These benefits improved insulin sensitivity and helped control blood sugar. But although the number of mitochondria increased, the efficiency did not. The electron transport enzymes in diet, exercise, diabetic patients were still only half as active as the same enzymes in healthy individuals. In June 2010, Mary Elizabeth Batty, a professor at Howard Medical School and researcher at the Joslin Diabetes Center, and Sylvia Corvera, a professor at the University of Massachusetts Medical School in Worcester, published a comprehensive review of existing research on the role of mitochondria in diabetes. They were forced to conclude that a defect of cellular respiration may be the basic problem behind the modern epidemic. Due to failure of mitochondria to adapt to higher cellular oxidative demands, they wrote, a vicious cycle of insulin resistance and impaired insulin secretion can be initiated. But they were not willing to take the next step. No diabetes researchers today are looking for environmental causes of this failure to adapt of so many people's mitochondria. They're still, in the face of evidence refuting it, blaming this disease on faulty diet, lack of exercise, and genetics. This in spite of the fact that, as Dan Hurley noted in his 2011 book, Diabetes Rising, human genetics has not changed, and neither diet, exercise, nor drugs has put a dent in the escalation of this disease during the 90 years since insulin was discovered.